All right, our next topic is about DNA replication, transcription, and translation. And I have a PowerPoint for this, but there's a lot of detail. And again, for those of you who are going on into a scientific field, I would hope that you would spend a lot of time studying this, a little more than you would need for the midterm or the final. The students who are not looking at a scientific career, you might want to take a very close look at this list of vocabulary words that I've provided on the page. So the video will be in between this. So below the video that will be inserted here when I'm done, there will be this vocabulary list that you are seeing here. You need to know these three major words about how DNA is copied, made, used, and it's replication, transcription, and translation. You need to know these three words. These are the keys, as you might guess from the title of this section: replication, transcription, and translation, keys to knowledge of this content. And then to understand these, there's another set of words: ribosomes, DNA, RNA, mRNA, rRNA. Notice there's a little lowercase r. And then tRNA. So that's messenger RNA, ribosomal RNA, and transfer RNA. Three kinds of RNA: codons and anticodons, and amino acids. If you know what each of these are in terms of defining these three words at the top, replication, transcription, and translation, you will do just fine on this section of the exam. So here is the PowerPoint. Before a cell can divide, the first thing it must do is replicate the DNA, duplicate the DNA. So replication is just a scientific term for duplicating the DNA prior to cell division. So you can see here in blue is the DNA double helix molecule. Looks like a ladder, and A's always go with T's, G's always go with C. So AT, GC, CG, AT, etc. It doesn't matter the order, TA going in the other order. It just is always A has to bind with T and G has to bind with C. The way the molecules are shaped, it's like a lock and key mechanism that uses special hydrogen bonding. The way they bond, they fit together like a lock and key. And that's what keeps the DNA molecule not only together, but also allows it to be replicated accurately. Because as you unwind the DNA molecule, wherever there's a C, a G must go. And if the next one's an A T must go. If the next one's a T, it must be an A, etc. So there's no question when you unzip a DNA molecule to replicate what goes next. And so you can see here in gray how special enzymes have come in to fill in the blanks. And these enzymes will keep adding until it's completely unzipped and you have a complete replicated DNA molecule. Each new DNA molecule will consist of one old strand and one new strand. These are called complementary strands because they're not identical. On this side, if it's ATG, this side has to be what? ATG, so this one has to be TAC. They're not the opposite. They're not a mirror image. They're called complementary. And because you have one old strand and one new strand, the process is called semi-conservative replication. There might be a bonus point. I don't remember if I'm going to do that this year, but there might be a bonus point to remember that. Replication is actually called semi-conservative replication, meaning one old strand is conserved and a new strand is added to it. So if I look at the two new strands that are made, it's not copying the entire DNA molecule, it's copying half of it twice. So all new molecules are 1/2 old strand DNA and 1/2 new strand DNA. So here's the assembly process. You can see here is the molecule all in blue on the left-hand side. In the middle, you can see it split. And so special enzymes come in, replication enzymes, and they match these lock and key pieces, the A's to the T's, the C's to the G's to the C's, the T's to the A's, etc., until you end up with two identical DNA molecules in their order. And it's a very, very fast and very accurate process. But there are mistakes that are made, and so when mistakes are made, there are other special enzymes, the cleanup crew, you might call them, that go through and they read the freshly made DNA, and if they find an error, they fix it. And even that isn't perfect, and that's how mutations can come about. But it is a very long molecule usually. DNA is long, there's lots of it. So it doesn't start at one end and go all the way down like a zipper to the other end. It actually replicates in these bubbles of start, and it goes in both directions. And it takes special enzymes. One set of enzymes is specifically designed to go left to right, and the other from right to left. And so you've got to pay attention to the detail if you're really looking at how this process works in a molecular way. For the class, I'm not too worried about it. Just understand that the DNA molecules are being copied, and they are being copied in a very efficient way. All of this is being done prior to cell division. We've already talked about mitosis and meiosis, cell division. What else do I want to put here? I'm not going to worry about anything on it. You can read that yourself. So once we have actually replicated the DNA, and we have a cell, now that cell has to function, it has to do something. So replication, that first word in our vocabulary list, is all about cell division. The next two words, transcription and translation, are all about using the information on that DNA. DNA is just a blueprint. DNA is an instruction manual for how to make a plant or an animal or a bacteria, anything. And so what does DNA primarily do? Its major function is to make protein. Protein are the workers of a cell. They can also be structural things for the cell as well. So some DNA makes structural proteins, and some of it makes functional proteins, proteins that do work. There's all these things going on in a cell. Well, cells don't have hands, they don't have arms and legs, they don't have teeth. How do they do things? How do they cut things? How do they put things together? We put things together and we work with our hands, our arms, and our legs. Plants, or cells, excuse me, whether it's plant or animal, cells don't have those structures. They have proteins, and proteins do work. They can move, they can twist, they can do all kinds of things based on the chemistry of proteins. So since DNA has the information, the instructions for making these proteins. And so to make these proteins, we call it synthesis. Synthesis is just to make something. So protein synthesis, protein making, involves two processes: transcription and translation. So now that we have a cell, it needs to do some work. We look down here, the purplish color is inside of the nucleus where the DNA is held. And inside the nucleus, this purple section, we must copy the instructions from the DNA. That is called transcription. We're taking the DNA information and we're transcribing it into RNA. It would be the same idea as if you were being asked to build a building and you had to take a walk over to the plan room where the plans are stored for making the building. You're like, all right, it's time to lay out the interior walls for this bedroom. So you walk over to where the plans are stored, downstairs in the plan room, on a big table that's laid out, and you go to upstairs bedroom, you look up the information on the DNA, so to speak. You look up the information and go, oh, this is how the walls are supposed to go upstairs. So you copy the information down. You transcribe the information from the plans, the DNA, to your notes, RNA. And that's all it is. You're just copying the information into another form that you can move to the actual work site. So RNA is just a copy, a complementary copy of the DNA that can now be moved to the actual work site. And once that RNA is done being manufactured, this is now a single-stranded form. It is now sent out of the nucleus into the cell cytoplasm. And there's actually a whole mechanism of worker proteins here, which we'll talk about in a minute. And they translate the information on the RNA into a whole new language. And that's why they call it translation, because here we're going from DNA to RNA. They use the same language. We're just transcribing it. Even though there's slight difference in a little shorthand, it is basically the same information. The translation is going from a nucleic acid language into a protein language, a completely new language. It would be like your workers are speaking Portuguese, but your plans are in Spanish. Would you have to translate that information? Yeah, you'd have to translate it. Okay. For many years, biologists referred to a, oh, I'm not, you can read that. There's some good information there, but I'm going to let you do that. So, now, what about this translation? There is a universal code called the genetic code. And the genetic code is written with only 4 letters, A, T, C, and G. That's it. There are 4 bases, nucleic acid bases, A, T, C, and G. And these bases are then read in groups of 3 in order to specify each of the protein subunits. Protein is just like a string of pearls, with each pearl having a unique amino acid. So we think of a string of pearls joined together, and each pearl could be any of 20 possible different types of pearls to make this beautiful necklace we call a protein. And each of those pearls is unique. Okay? So to go from 4-letter code into a 20 group code, that's why we call it translation. It's a whole different language. And so this code is based on groups of 3. And if you look at this illustration here, you can see where they say codon. This is the group of 3. U, U, U. What the hell is a U? Get to that in a second. G, G, C. C, G, U. U, U, U. Again, all of these are groups of 3. And the message is read in a group of 3. Okay, and I'll walk you through this in just a second. The message in the DNA is transcribed into RNA and then translated into a polypeptide. And a polypeptide is just a short string of a protein, of amino acids that are a protein. We first call them polypeptides because they're just the pearl necklace. When the pearl necklaces are all assembled into the finished product, the finished product is actually called the protein. And it could be multiple peptides making up one protein. Proteins are the most complex of the units. It's been processed, it's been assembled, it's now ready to go out and do work. So let's walk through this illustration to understand what's going on here. We have our DNA molecule, and along the DNA molecule, there are certain coding regions and non-coding regions. The coding regions we call genes. And the version of the gene is called an allele. So if I have a version of a gene, so let's say an eye color gene, the version that I have, the allele that I have, gives me brown eyes. Now you might have green or hazel or blue eyes. Now it's not just one gene that gives eye color. Eye color is more complicated than just one gene, but a gene is just going to code for a protein that does something that causes a result in the finished organism. The version of that gene is going to determine what that result is. So a flower color gene, maybe you've got a flower color gene and one allele gives you white flowers, another allele gives you pink flowers. Same idea, all right? So now it's time to do work. The cell says, hey, we need information from gene number 3. And order goes in. We need information. We need the protein from gene number 3. So gene number 3 is first opened up to expose the 2 sides, so it's unzipped, and then it's copied into RNA. Now, RNA is a little different. It's not deoxyribonucleic acid, DNA. It's RNA, ribonucleic acid. Doesn't have the deoxy part. Because of that, there's a slight variation in the base for thymine, and there's no T in RNA. It's called uracil, U. And so don't get confused. It's just everything else is different, except when you're talking about RNA, any T's become U's. In DNA, the U's are T's, okay? That's it. It's just a substitute. The T and the U, they're the same basic thing with a minor difference. They still have exactly the same lock and key mechanism. They still fit exactly the same, but it's called uracil in the RNA form, all right? That is one point of confusion here, but it's not that difficult. So you can see how it's being transcribed. A's would normally go with T in DNA, but here there is no T, so it's U, 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 G, G, C, and you're just copying the opposite. A goes with U, C goes with G, G goes with C, and the only one they don't show here, if there was a T on the DNA side, a T would go with NA on the RNA side. T with A, because there is an A on RNA. And then Now these codons, these groups of 3, are read one group of 3 at a time to determine which amino acid goes there. All right? And that is the genetic code primer. We'll talk about this more in a moment.